what I will do is present you today some recent work on altered innervation in cachexia. So I will do, of course, a little bit of introduction, even though you don't need much. I will try to go to put this in the context of some problems of regeneration in cachexia, and then I will go into the denervation thing, and it's still a controversial issue. So I will do a little bit of discussion, hopefully with you, but with the aim to convince you that maybe cachexia should be started to be regarded as a neuromuscular problem, not just a muscle wasting thing. Anyhow, this is the question we start from. That is, are all wasting conditions the same? What happens in cachexia, even though very likely this is fasting? But what happens in young people that are losing weight in respect to old people that are losing weight? Well, definitely these two conditions are similar in some aspects, because in both cases they are characterized by wasting, muscle wasting, and an impairment of the function of the musculature and its metabolism. And this is likely due to dysregulation of the humoral factors and the local factors affecting muscle homeostasis. In both cases, these conditions are associated to increased morbidity and, of course, increased mortality. Remember that about let's say 20% of cancer patients die of cachexia, not of cancer. And of course, 100% of very old people die. So that's also a very bad prognostic condition. The two conditions, though, are quite different if you look at the histological level. So they also have differences. Cachexia, for instance, is characterized by a generalized muscle fiber atrophy, while sarcopenia can display this selective fiber atrophy that correspond to angular fibers that are squeezed between the others. And usually these are the fast fibers. And this is the change and the change in fiber type and the selective that you see here. The increase of the slow fibers and the grouping of these fibers is likely due to alterations in the innervation status of this muscle. Overall, all these changes lead to a lowering of the musculature, in fact, of the person, and to weakness, and to also a lower coordination capability. All these neurological, let's say, changes are totally unknown in cachexia. Nobody knows what happens to nerves and stuff like that in cachexia. Nonetheless, what is well established is that in cachexia, we definitely have a loss of muscle function, lower strength, muscle strength, lower ability to perform kind of muscular activity that is also kind of subtle, and you need coordination and equilibrium and so on. This is associated. Sorry, I need to remove. Well, forget about it. This is also associated to a decrease of a specific force of the musculature, meaning that the muscle is specifically affected. And we also have contributed to the field by showing that the fatigue is increased. Cachectic muscle in tumor-bearing mice, C26 being a tumor, are characterized fatigue more rapidly. And what is observed in the murine model is also true in humans. In fact, the group of Kizari, for instance, one of our colleagues and friends that usually follow these meetings, show that there is a decrease in force in cancer patients, and also a decrease in the resistance to fatigue. So they lose the capability to contract the muscle in a more significant way. And the final result of all this is that cancer patients move less, probably because they fatigue more. And this is a progressive loss that correlates with the worsening of the pathology. So there are very important consequences in the loss of muscle activity and function. And we all know in this audience how important is the neurological control for the muscle function. We have heard that in all the talks. What I would like to stress is not really this very basic part of the slide, is the fact, as shown by Professor Miyake, for instance, earlier today in his beautiful presentation, that the muscle fiber is such a big thing that cannot just die and disappear if it's damaged. In many cases, damage is repaired, and there are immediate repair processes, as shown by our Japanese colleague today. And also after that, to fill the gap between the stumps, there is the intervention of satellite cells. Professor Musaro has already done a very nice introduction this morning on the inflammatory phase, so I will skip it, and I will stress the fact that when the myoblasts fuse, align and fuse, they form these strings of nuclei within the nascent myotubes. We call them, of course, myotubes because they have this form. And so these central nuclei are very peculiar morphological features of regenerating fibers. And this is something very similar to what happens during embryogenesis. All of us in our classes, we tell students that regeneration recapitulates the development. Um, the, the, the reason why these nuclei are in the center of the fiber is not so clear after all, but definitely it's something that is tightly controlled. This is a scheme from cell cultures. The nuclei are added at the periphery of the nascent myotube, but it's also kind of true in vivo. Nuclei are added to the nascent myotubes. They go into the center of the myotube, then they spread, and each of them occupies a certain nuclear domain, and only at the end they go to the periphery, and sometimes, in some cases, actually, they group in specific regions, such as in the underneath the neuromuscular junction. All these things has been well characterized in terms of mechanics and the cytoskeletal components. What I would like to stress again is that even if a nucleus starts from the periphery of the to be added to the myofiber from the periphery of the myofiber, it goes to the centers and then go back to the periphery. So all this waste of energy and mechanism must have an evolutionary advantage that is still not really known. Otherwise, it would not exist, of course. Another feature of regenerating muscle fibers is the expression of the developmental myosins, such as the embryonic and also the fetal and the postnatal myosins that are not that are expressed in the embryo and then in the fetus. Of course, their meaning again it's kind of object of speculations. Nobody really knows which are the reasons why embryos have different myosins, and also it's not known why these myosins are expressed during regeneration again and not the adult myosins directly. There must be a reason. For now, we just speculate and we use them as a very useful markers of muscle regeneration. Uh, Last but not least, of course, I want to cite Helmut's and Hugo's and the others' work showing the presence of central nuclei in a totally different condition. This very atrophic muscle fiber is due to denervation, and the nuclei are central. And again, we have this weird expression or re-expression in the adult of embryonic myosin isoform, and there must be a reason for that. So now at this point, I think you're convinced that central nuclei are a clear sign of myopathy. If you see a central nucleus, there is a damage. There has been a damage before. That's true both in acute condition or in chronic conditions. And these central nuclei so are a marker of presumably of damage and regeneration. By the way, I call them central nuclei. And let me just take advantage of this presentation to advertise a little, not only to advertise an opinion article that I wrote, but also to tell you that when I wrote this article, I realized that if you do a search, PubMed search for central nuclei, or a very similar expression that is centrally located nuclei, almost the same thing, you find totally different groups of articles. So be aware of this. If you're interested in muscle regeneration in central nuclei, change the expressions because different groups use different vocabulary for the very same thing. This being said, as I showed you, the central nuclei are present in pathological condition. Of course, this is a different thing. This is a fiber that is engulfed and invaded by macrophages. And then there are these conditions such as cachexia that do not really show overt myopathy. I mean, not many not particular signs of central nuclei in the fibers. However, if you look carefully at these cachectic muscles with a specific dye for damaged fibers, such as these Evans blue dye, you do find, as we did, some damaged fibers that release creatine kinase that is increased in the serum of these animals. So there is muscle damage in cachexia. And the reason why probably comes from a study from the Guttridge lab showing that in cachexia, for instance, in a cancer patient, dystrophin, it's almost disappeared in respect to control patients. That means that a little bit like in dystrophy, but this is not a congenital genetic condition, it's an acquired condition. These patients are almost dystrophin knockout, sorry. Therefore, or knockdown actually. So the muscle is fragile and then it's damaged. We do expect to see muscle regeneration after damage, of course, but that's not the case in cachexia. If we do an assay doing a focal injury and we follow up the regeneration process that occurs normally in control animals, and we compare this to tumor-bearing animals, we see that there is a persistent mass and inflammation keeps going, and the regenerating fibers are not there or are greatly reduced. And we also did the reversal. Sorry, and this is not due to lack of myogenic capacity. If you remove cells from this abortive regenerative muscle and you put them in culture, these cells from cachectic animals are able to differentiate even better than the control cells. So it's not that the cells are lacking the myogenic potential, it's really the environment. And we did the reverse experiment. We injected in a mouse, an nude mouse bearing a tumor and injured. We injected cells that we could follow because they were like Z, so they were blue, and they were perfectly happy and able to make myotubes in culture. We injected them, and we noticed that in the control animals with no tumor, these cells participated and fused, participated to the muscle fiber that became blue, while in the tumor-bearing animals, they just remained, survived in the interstitial space without fusing. And we demonstrated that this is due to an increased level of PAC7. Basically, these satellite cells remain kind of activated myoblast expressing PAC7, MyoD, etc., but they do not terminally differentiate. Now, let me really go to the point by starting with this observation, very interesting observation by Sandra Zampieri and Ugo and all of these friends and colleagues on cancer patients at an early stage. So this is an early event, and they have observed fibers with central nuclei. And this observation was kind of unaccounted for. That prompted us to try to find a reason for these fibers. First, we really wanted to get confident with the use of these markers such as nuclei and embryonic myosins. So we did some preliminary experiments in mice, and we did the kinetics of these markers in mice that are healthy, no tumor, just an injury. And we followed the expression of embryonic and neonatal myosin and the central nuclei that you see highlighted here by the arrows. Sorry if they are small. Basically, we noticed that the central nuclei are very high after injury and remain very high. It takes a long time to disappear, while the molecular markers, the myosins, disappear much in a faster way. They last only for like 9 days, 10 days, okay? So then we did not do the injuries. We went to look to the tumor-bearing mice. So in the absence of experimentally induced injury, just the tumor, we looked at the muscle, at the tibialis of these mice, and we noticed central nuclei in the fibers. And this number was increased in respect to the control, significantly increased in the tumor-bearing mice. And this was always in the absence of other molecular, let's say, regeneration markers. So the two things were uncoupled. And if you remember the kinetic of the expression of these regeneration markers, here is where we do the stop of the experiment and we do these measures here. It's 19 days following tumor transplantation. This is the time that the myosin are expressed during regeneration. So if any expression occurred, it would have been in the very first day of when the tumor is just implanted, not even vascularized, and it's not really felt and not doing its effects in the mice. So basically, we are quite confident that you can have central nuclei in the absence of markers of regeneration. And these nuclei form clumps, exactly like in the denervated fibers that I've showed you before, and the number of nuclear clusters increases. And these are not these clusters are not due to let's say macrophage nuclei, because they are really in within the myosin that is shown here in red. So really, where there are the sarcomeres. Okay, so it's not just an inflammatory invasion in this case. We found this kind of interesting. So we also looked at denervation markers. And as Sandra has shown you, NCAM is a marker of denervation. This is our positive control. Its expression increases a lot. In healthy mice, you see this expression only in satellite cells, but you see appearing NCAM positive fibers in the tumor-bearing mice in the very same muscle that was showing the central nuclei. So we wanted to extend this analysis, and we looked at another muscle that was the rectus abdominis. Again, we scored for central nuclei. We noticed an increase in these fibers with central nuclei, and in the very same muscle, extract showed the molecular analysis showed an increase of a plethora, a global increase of a plethora of different markers for denervation, such as acetylcholine receptor, again, NCAM significant in this specific case. Histone deacetylase was also significant increase, MuSK, etc. Why did we choose the rectus abdominis? Because in spite of the anatomical and species differences, we had the possibility to have a look to the rectus abdominis from human patients, thanks to our collaboration with the University of São Paulo in Brazil. And the interesting thing is that in this case, not only it was humans, so I mean, very interesting to us, but also we had the three groups, not just the two groups, not only the control and the tumor-bearing patients, but also we had two subgroups. On the right, you see the histology and the measures of central nuclei of patients that were having a tumor and displaying cancer cachexia. They were really cachectic. On the left, in the center, let's say, of the image, you see cancer patients that were weight stable. They were not suffering from cachexia. So the only difference here is the really cachexia. And the significant increase of the central nuclei in the myofibers correlates with the cachectic states and with expression of the denervation marker. So I hope that in agreement with Sandra's data, I'm convincing you, hopefully, or at least start you to stimulate you about this idea that might be problems of innervation in the cachectic muscle. The story is controversial, though, because while we were doing this observation, another group reported in a very good journal, unfortunately, a negative result, that it's the opposite of what we said. They showed, though, a negative result. It just showed neuromuscular junction in cachectic muscle. Whether this is a false negative result, I mean, they don't have a real control in the sense that, maybe they have missed them. I don't know, but that's what they published. So I have to report that. On the other hand, there is our reports. There are these data from Sandra and other people and Marco Sandri that clearly some stuff of this is still unpublished and struggling for publication, clearly showing that there is denervation in cachexia, and in agreement with basically all reports showing the occurrence of these angular fibers that are a histological marker of motor neuron death in patients that are cachectic. So as you can see, there are the two parties here. Our contribution, though, is that definitely there is an alteration of innervation, and it's clear that cachexia is also characterized by disturbance in the excitation contraction coupling. And this might do, as Feliciano nicely show, this is the territory of Feliciano now for new research to see whether these alterations that he shows are present in cachexia as well. And also, I think it might be due to altered innervation. So what we propose, what I wanted to tell you today, is that there is this, in cachexia, there is this uncoupling that it was not observed until recently between increased muscle damage on one hand and hampered regeneration on the other hand. This contributed to the imbalance in the muscle homeostasis. On top of this, in both cancer patients and animal models of cancer cachexia, we observe displaced nuclei, and the regeneration, it cannot account for these displaced nuclei. We don't see any marker of regeneration. We have shown, we and other people, many other people have shown that there is a blockage of regeneration in cachexia, so it cannot be just regeneration. So they might represent a previous neglected phenomenon of altered innervation that is occurring in cachexia, leading to a displacement of the nuclei from the subsarcolemmal position to the central one. And so my proposal and my stimulation for the discussion is the following. Maybe from today we should start thinking regarding at cachexia as something that is not just a muscle wasting syndrome, but more kind of neuromuscular problem, and we should act consequently in the therapy and in the medical approaches and supportive approaches against cachexia. I will stop here, thanking, of course, the two groups. Well, the groups are really mixed because we have plenty of students that are in co-tutels, so they're co-tutored between Sapienza University in Rome and Sorbonne University in Rome. So I thank especially Mehdi, who was a PhD student who did this job, and Nisreen, who was a postdoc, and then the people in Brazil, especially Gabriela and Raquel, that are postdocs in Marile Silender's group that did the human analysis, and of course Marco Rocchi, that is always with us for the statistics, and he's at the University of Urbino. So thank you very much for your attention.
Dario, looking at your results, it would seem that regeneration is not even probably a problem in the humans in cachexia, because central nuclei are very rare in humans anyway, and certainly after regeneration, they don't stay very long. So it would seem more that there's something other than regeneration to explain the central nuclei, which would be in favor of your second hypothesis.
Okay, thank you.
Oh, sorry.
No, go on.
No, just finish.
You have a question?
Yes, I have a comment and a question. First of all, thank you for the data you have shown, also because are sharply in accordance to our data. And I have a comment on the Gillingwater paper in which they have demonstrated that the neuromuscular junction in cachetic patients are stable. They did analyze only muscles from patients affected with esophageal cancer. That is a cachetic cancer, of course, I mean a cancer that induced cachexia, but it seems that not all the same type of cancer induced cachexia and muscle wasting in the same way. Our observations were obtained from pancreatic cancer and colorectal cancer. So therefore, this could be the reason of the differences we observed. But we have also esophageal cancer patients, and we are starting to analyze those as well to see whether Gillingwater is right. But one of my questions to you is regarding the myonuclei that I and you found within the myofibers, since there is a denervation issue. So do you think that some of those myonuclei can come from the neuromuscular junction, that are some of the myonuclei belonging to the neuromuscular junction or not? Did you try to stain those myonuclei with specific markers for nuclei of the of the neuromuscular junction
Yeah,
to
thank you,
test this
Sandra.
hypothesis.
Thanks for the comment. Yes, of course, I also wanted to discuss the story of the seemingly or apparently contradictory reports between different groups. Definitely, we are at the beginning of this observation, so of course, everybody noticed something. There is likely some aspects of the whole of the big pictures, and one day we will need to reconciliate the different things. The different views. So we will see. For now, we report all the observation and we put them in the discussion, and then we will see, hopefully, to have hints to understand why there are these different observations. And for the nuclei, sorry, but we didn't not label them. Of course, there might be some work going on, especially also Costelli in Torino is interested into this, I guess. So we will see in the future. But yeah, I cannot really tell the origin of this, the original position of this nuclei. Sorry.
Thank you.
Okay, I just want to remember that central nuclei are seen in many conditions, like central nuclear myopathy, myotubular myopathy, myotonic dystrophy, and so on. So, I mean, we don't know what keeps the nucleus to the center or what sends it to periphery. In the heart, there is nothing strange to see a nucleus in the center, but obviously not in the muscle. So it's a mystery. But I see that there was a question from Adamo. Is he still willing to ask?
Yes, please. Dario, thank you very much for this presentation. Very interesting. I want to say to everyone that we didn't agree on this, and so it's just a real question. It is.
Why you didn't agree?
No, we didn't agree on the compliments in advance.
The paper that was up.
No. Is it possible to establish a relationship, except for the most obvious and most extreme cases, between the presence of not peripheral nuclei, let's say central nuclei, and the ability of the fiber to contract? That is, is it, let's put it in another way, is it possible that the movement of a newly added nucleus from the center to the periphery of the fiber is influenced by the contractile activity of the fiber itself? And do you know whether anyone has ever studied this, and whether it is possible to study this?
Well, the person who is all the best studies, who's doing the best studies on this is Gomez. In my opinion, of course, he was now back in Portugal and was with Gilles and other people in Paris, and basically they have contributed to show Edgar Gomez, I don't know what I said. They contributed to show very clearly the mechanism and all the proteins that are involved in nuclear movement. Then, of course, your question that is more generic, like is contraction affecting this movement? So behind the specific players that really literally transport or anchor the nuclei somewhere, does contraction affect this movement? This is a very interesting question, especially because maybe, in the embryo, the movements are different from the adult, and, but I don't think anything is known. Maybe Jill, maybe she has some.
No, it's still an open question.
It's a very interesting question, though.
Very interesting. There's a lot of people working on it.
Thank you. Okay,
well, I
we'll discuss about it.
Ugo.
Let me say that
Ugo, turn on the microphone. Switch on the microphone.
Who counts my microphone? Let me say that the traditional explanation of nuclei, of central nuclei, is block of differentiation, They are there because the transport to the periphery is not possible, not
Well,
realized.
that was the concept of myotubular.
This is the what do you find when you look at the literature related to neuromuscular genetic diseases?
Yeah.
Block of differentiation.
Yeah.
And therefore the nuclear. But there is a very strange cases. If you do classical muscle damage and regeneration by toxin, venom toxin, or high anesthetics, there are strains of so-called normal mice that do not complete the regenerative differentiation. You know something about this, Gillian?
No, because usually using any of the toxins, the muscle regenerates in the mouse, but the nuclei stay central for a large number of months, and they eliminate the developmental heavy chains. So
there are strains that the normal, considered normal, that have some defects, somewhere that is unable. Another problem is if the nuclei move from outside to inside. And this could be, it is very, very difficult to demonstrate what happens to those nuclei if they do not went out or were moving from the periphery. What is sure is that there are molecular motors and mechanisms continuously active during the life of the animals, of the muscle fibers, who maintains there the nuclei.
Yeah, that's for sure.
They say
because
DNA can be absent.
Very, any trauma, any simple damage to the muscle fiber, the nuclei are displaced.
It's a complex phenomenon.
Why? I don't know. I know that is for sure.
It's a complex phenomenon. We have to stop the discussion at this point. Next year will be one of the issues that we'll discuss: why nuclear center in certain disease, why they are in Cachexia, what about genetic myotubular and whatever centronuclear, but that's a different story. So we have to carry on.